Hi, I'm Dharanija Madhavan. I'm a scientific coordinator at the Max Planck School MtL Live, and it's my pleasure to have Dr. David Zwicker, who is a fellow at the MtL Live network since 2021, um, join us in this MtL Live lecture series, which is held every month on Wednesdays, generally at 3 p.m. German time, where we have a fellow each month discussing about their research within the scope of MtL Live. And this month we have, as I said, Dr. David Zwicker, who did his PhD at Max Planck Institute of Physics of Complex System in Dresden under Frank Jülicher, who is also a fellow with us. And he joined the group of Michael Brenner in Harvard to do his postdoc. And since 2017, he has been an independent research group in the Max Planck Institute for Dynamics and Self-Organization in Göttingen. And his research focus is physics, physics of biology. So it's extremely relevant to MtL Live, and particularly he uses statistical physics, dynamics, fluid dynamics, and information theory to explain biological phenomenon and tools such as this. So we're happy to have you. Thank you so much. Before we start with the talk by David, we would like to ask two questions to set the stage. So the first question is, can you tell us about your current role at MtL Live and how your work contributes to this research field?
Yes, of course. So as you already nicely introduced, I'm a research group leader, meaning that I spend most of my time doing actual research, fundamental research, leading a research group comprising postdocs and PhD students mostly. But we also have a fair number of internships through the MtL Live program here. And so to really put forward our interdisciplinary research, we try to understand various biological processes through the lens of physics, really. And so we are developing theories. We have a theory group to describe complex biological processes, basically unveiling the physical laws of living matter. And we sometimes model concrete experiments, but other times try to really get at the bottom of some theoretical description. And so we live in this broad scheme, as I will also make hopefully a bit more clear with my talk.
Okay. And our second question is, what is the most significant or are the most significant challenges you've encountered in your career, and how did you address them, and what advice would you give to students who want to take this path?
Well, I think in this interdisciplinary research that we and many in the MtL Live program conduct, one of the key challenges is simple communication. And that's because we come from different backgrounds, we learn different terminology, and it's very hard to communicate ideas across disciplines. And I first encountered this actually during my PhD in Dresden, where I was in a theory institute, but I also collaborated with Tony Hyman, who I think is also a fellow in this school. And he leads a biology group, and I actually spent one day a week at their institute participating in the group meeting. And I honestly didn't understand anything the first year or so, simply because I didn't have the necessary biology background. But I think it's absolutely paramount to establish a common ground and common terminology to be able to communicate. So I basically worked hard, went there, talked to people, talked to students in particular who helped me to understand what they were doing, and slowly learned their language, which helped tremendously in communicating, understanding what they were actually doing. And it also forced me to explain my work in simple terms that biologists could understand. And I think that's really key. That's also something I think where MtL Live excels at, training students early in this communication aspect. So I think that is the main thing I want to mention here.
Thank you. So before I hand over the stage to you, I would like to tell those attending that we have a Q&A button at the bottom. If you have any questions, please type it out in that, and I would ask David at the end of his talk, and we will go through the questions and they will be answered. So with that, I look forward to your talk.
Well, thank you. So I'm excited to be here and give this talk in the MtL Live series. And I decided to name my talk Droplets Come to Life, fitting with the matter come to life. And then droplets are just one way of matter, I guess. And there are two terms in the title that I'd like to clarify. But before we go there, I would like to start by acknowledging the people who actually do the work. And so here I show you a slightly outdated picture, which is now 2 years old, of our group. As I said, comprising mostly PhD and postdocs, but actually also 2 MtL Life students standing in the front row here. They are actually pivotal for keeping the group fresh and bringing in new ideas. And here you have a list of the current PhDs and postdocs, which are mostly theorists, mostly physicists actually, because that's our core expertise. But as I stressed multiple times already, I focus a lot of interdisciplinary research and communication. And the way we achieve this is by collaborating with other people from around the world. And this is a list of current collaborators comprising various disciplines and also theorists and experimentalists. All right, so I already said that I want to talk about droplets, I want to talk about life, and to bring everybody on the same page, I'd like to define these two words first. And so let's start with this very broad question of what life actually is. And you might have heard about this question, in particular if you did research for MtL Life. And I think what often comes to mind, at least when you're a physicist, is this little book by Erwin Schrödinger, which is exactly named What is Life? And I think it's a marvelous book. It's really interesting. But I still like to maintain that the question that he is raising, and I'm also raising the title of my slide, is actually a slightly weird question. It's in some sense a wrong question because it's hardly ever answerable. And the reason I think this is the case is because it's a qualitative question. It basically maintains that there's something that is life and something that is non-life, and there's a boundary that we can define. And I maintain that it's very hard to find this boundary because everything is blurred, everything is continuous, in biology in particular, in such complex systems. So what I propose to turn this into something that one can do research on, I would turn this qualitative question of what is life into a more quantitative question, and basically asking how much a particular system that we study resembles life. To make this even more concrete, I'd like to start by listing certain features that we demand of life that also come up in this book. So I like to start to set the stage by mentioning characteristics of life that I think are absolutely paramount to be there, which we could then set out to study and quantify. And I think one of the main things, and probably the first thing one would think about, is that life sort of transforms the environment. It uses energy, uses metabolites from the environment, converts them to maintain itself, right? And that one could probably describe as metabolism. So to convert energy from the surrounding, be it photosynthesis and using energy from the sun or some chemicals, you need energy to maintain function. Another thing that is very characteristic of life, and I think very interesting from a physical point of view, is what one could call homeostasis, which is simply the fact that living entities maintain their state. They basically ensure that they don't deviate too much from a state in which they're happy. So that has to do with stability, and is directly linked to metabolism, because from a physical point of view, they use driven processes, they use energy fluxes to maintain their state. But of course, life is not only stationary and maintained fixed over time, but it can also develop. In particular, it starts growing typically from a very small cell to maybe a multicellular organism. And so in some sense, it's able to adapt to the environment, it can grow, it can change. And the tension between homeostasis and development, I think, is very interesting and productive if you want to think about what life actually is. And then it's clear that in order to maintain life, because there's death, there also needs to be a way to create new life. And that, in all cases that we know of, happens through reproduction, meaning that life can duplicate, replicate. And one interesting aspect that is, again, from a theoretical point of view, very crucial, is that life can pass on information, mostly in the form of DNA, but there are also some other forms to pass on information. And so that also needs to be a part of the definition of life, I would maintain. And something, a fifth point that I would like to stress has to do with response to the environment, environmental changes, because the environment is fluctuating, it's changing over time, be it seasonal changes, be it daily changes. Life better has a way to adapt to these changes. And this one could characterize as a response to stimuli. And the last thing I want to list in this list here is that life needs to change on long time scales. It needs to adapt to, let's say, climate change, for instance, through evolutionary adaptation. And this one could also categorize a response to stimuli, but I would separate it out because it ties in with reproduction directly. And from this small discussion that I'm bringing forward here, you can already see that this list is by no means exhaustive. It's by no means the only list you can make. And even the categories I put forward here as examples are not super clearly defined. They are connected to each other, like reproduction, evolution. And I think this highlights how difficult it is already to define life, and also highlights how multidimensional life really is. Because I think, and that comes back to the original question, I think it's not sufficient to just ask this single question, but rather view life as a multidimensional process, a multidimensional phenomenon. And we can now set out to quantify how much a particular system resembles life along these dimensions. And that's really what I want to do for a particular example in my talk. Now, one thing that I've left out from this definition, because it's probably so fundamental that I put it separately, is that life also uses a fundamental, there's a fundamental unit, because life needs to be contained, self-contained in a particular part in space. And for all forms of life that we know of, all of biology, this fundamental unit of life is the cell. But one could presumably think of other fundamental life forms. And that's indeed what I would want to bring forward in this talk. Because if you remember, my talk also had the word droplet in the title. So we need to also discuss droplets. And what are droplets? Well, this is arguably easier to answer because everybody's seen droplets before, right? You just look at a window pane, if you showered or whatever, and then you have droplets precipitating there. Now, I'm a physicist, so I'd like to not just show pretty pictures, but actually give you concepts of how one can think about droplets and the physical process of phase separation that tells us how they emerge to put basically everybody on the same ground. So in the simplest case, you can think of droplet formation as a physical process which somehow brings together molecules in a small space. And this has certain requirements, which I want to lay out here. And to do this, just think of a box. Here, this gray box, this is your system, conserved volume. You have some molecules in there. And in the simplest case, if these molecules don't interact, they just buzz around and mix evenly by diffusion, basically. So that's what you would expect most naively from an ideal gas, for instance. Now, in order to have droplets, you need some additional process. You need some attractive force that want to bring the droplets together. And this could be some weak multivalent interaction of these molecules so that the entropic loss of concentrating the molecules in the small volume is offset by an enthalpic gain of these interactions. So basically, the few molecules that are in this large dilute volume here, they enjoy a lot of entropy, but little interaction. And conversely, the molecules in this dense droplet phase, they are confined. They have not a lot of translation entropy, but they enjoy all these interactions and thereby gain enthalpy. And from this entropy-enthalpy balance, you can already see that there is actually an interesting part of the system which is, so to speak, not quite happy. Namely, when you focus on the interface here, the molecules there don't enjoy the entropic freedom of the dilute phase, but they also don't have as many interaction partners as the molecules deep in the dense phase. And this leads to an energy penalty of actually making an interface, which we commonly call surface energy or surface tension. And so systems that tend to lower the overall energy, the overall free energy, then would want to minimize this interfacial area and the interfacial energy. And that's precisely the reason why droplets are typically round, because that's the geometry of the lowest surface area for a given volume. But this surface tension also has an additional effect, which comes into play when you look at the multiple droplets. So here I'm putting a schematic where I have one large droplet, one smaller droplet. And you can imagine that this configuration has a larger surface area than if I put all the dense material into one large droplet. And indeed, the surface tension is such that it induces a flux of molecules from the small to the large droplets, so that eventually the small droplet dissolves, the large droplet grows to actually reach this stable equilibrium state. So what I'm saying here is that surface tension makes droplets round, but it also leads to an unstable situation if you have multiple droplets, all the small droplets disappear and the large droplets grow. And this unstable situation, this leads to this coarsening process of smaller droplets disappearing, larger droplets growing, which one can actually quite easily simulate using this seminal Cahn-Heald equation. I don't want to go into the details of this, but this is basically the simplest equation which can describe phase separation in a concentration field C. I just want to show here what happens if you put this equation into the computer and simulate it, and then you see with the right initial conditions that many, many droplets can form. And if you look closely, you will see that all the small droplets disappear and the large droplets grow, which is precisely because of the surface tension that I introduced. So this is all I wanted to say for the physics of phase separation. So basically it tells us how molecules come together to form these droplets. And now I want to connect this physics of phase separation with this idea of all these aspects of life that I mentioned earlier. And one thing that I would like to start with is to basically posit that these droplets themselves already form a well-defined entity. So in some sense, I want to replace the word cells that we traditionally think of as the fundamental unit of life by the word droplet. And I try to see how far I can push this analogy and try to ask, how much do droplets actually resemble life? And could they be some early form or fundamental unit of life? So the game of the talk today, next 20 minutes or so, I basically want to ask whether droplets in the physics of phase separation can fulfill, or how much can they fulfill, each of these 6 categories that I introduced earlier. Okay, so let's just go through this step by step. And one thing that we already need when you think about droplets as a form of life, in some sense, as a more theoretical endeavor at this point, is we need a collection of droplets. We want many droplets. We want many droplets of the same kind so that you can actually have a whole population. We might also have multiple types so we can have evolution. So the first thing I think one should think about is how actually multiple droplets can coexist. And there are various different answers and various different ways. One simple way would simply be to just put them far apart from each other. You have multiple droplets. If they are far apart from each other, diffusion is so slow that they, to all intents and purposes, don't interact. Fine, but then you need large space and everything is slow and so on. That might not be the best way. There's another alternative for how you can stabilize multiple droplets. And that's simply by appealing to many, many different components. So far, I only talked about this one molecule that phase separates from the surrounding, but you can easily imagine, particularly if you think about a cell, that there are many, many different molecules that all interact slightly differently. And so you can make droplets of different composition, which might not mix. And in fact, one can make this statement precise. There's an upper bound to how many different kinds of droplets you can make in a thermodynamic droplets. That's Gibbs phase rule, devised some 200 years ago or so. And it simply says that you can't make more droplets in an equilibrium situation than there are components. We looked at this recently in a publication which came out late last year, and we simply asked how many kind of phases, kind of droplets you can make as a function of component count. And we found that you actually can quite easily get close to this Gibbs phase count. Here we looked at an example where you had random interaction matrices just to approximate the complexity of actual molecules. Now, what we found to our surprise is that if you make add more and more components to the system, which is on the x-axis here, the homogeneous state where we just ask everything to stay mixed becomes more and more stable, which is quantified by this orange curve, pink curve. I don't have time to go into the details of the analysis here, but what this is saying is that if I ramp up the component count, approximating a very complex mixture, I expect to find more and more phases in the thermodynamic equilibrium, but I also expect a mixed state to be fairly stable, which sounds contradictory, unless you think about it as a complex energy landscape where I have many possible minima that all metastable, and I can make many different states. And I try to illustrate this in this example here where we chose an 8-component system. This is a particular interaction matrix. Details don't matter so much. But then we look at spatial distributions of this material, of these 8 components. And numerical simulations showed us that both the homogeneous state, but also various other states are stable. meaning that even without using any non-equilibrium physics, we can already have a situation where multiple droplets can be stable, they can be created, they can be removed. So this is a big, complex playground that we can exploit. So that's nice. However, in all these situations, because we have this offset ripening and we want to minimize the surface area, you can only have one droplet of a kind, which is not what life would work with, because there you have large populations. So we need to do something else to stabilize droplets against this offset ripening that are introduced. And one particular mechanism that I would like to single out here, we discovered some 10 years ago, and this actually uses already one of the first characteristics of life that I introduced earlier. Namely, I can combine phase separation with a simple form of metabolism building on chemical reactions. And I will show you now that this can actually suppress offset ripening and stabilize multiple droplets. So here's what I envision. I have a droplet comprising some droplet material, which phase separates, much like I introduced before. And what I want to add now to this schematic is some simple form of metabolism, meaning that the droplet takes up material from the outside, some material influx, and has some internal reaction, some internal conversion, where in this simple case, I convert the droplet material to some base material W. That's just the simplest schematic I can think of. And this base must then leave the droplet. And to close the whole system and actually to maintain this over time, we then need also a replenishing reaction, which converts the waste back to some droplet material. Now, this is external to the droplet. This can be done by various different means. But I'd like to stress here that I'm thinking of a system which then uses energy to convert the low energy waste product back into a high energy droplet material molecule. And this could be driven by light or any other energy source. And if we do that, we can, for the simplest case, basically combine the Cahn-Hill equation that I had earlier with some reaction term. Again, I don't have the time to go into the details here, but you have to take my word that this is one way to mimic the chemical reactions that are graphed here on the left. And if I now simulate this case with exactly the same method, like the one I showed offset ripening for earlier, then we again see droplets. That's because we still have this phase separation part, which is described by the first part of this equation. But you might already see here that the dynamics are now very different, where all of a sudden the system goes towards a state which is very regular, in particular with maintained droplet sizes. And this is exactly what I was saying earlier, that this simple form of metabolism can suppress offset ripening. Now we can analyze this a bit more. We can, for instance, shortcut the replenishing reactions if we just have two conversion reactions. And the reason, in short words, why this is stabilized is simply that the influx of material is diffusion limited because the droplet is small compared to the environment. And that means that the total flux is proportional to the radius of the droplet in the 3D system, whereas the metabolism here is simply proportional to the volume of the droplet. That's the simplest assumption. So it grows with the cube of the radius, and the droplet size, the change of the droplet size is given by the net flux, the difference of the two, and then you can convince yourself that if the droplet is small, the influx dominates, so it grows, and if it's large, then the efflux dominates, so it shrinks, and in between there's a stable stationary state, which you can also analyze numerically. We've done this in a few papers. Now, details don't matter so much. The question is, does this idea have any merit? And I'm happy to say that basically last year, another MtL Life fellow, Jörg Böckhofen in Munich, finally succeeded to reconstitute this system in vitro. Here is some idea. Of course, realistic systems are more complex than what I showed in my schematic, but it has this interesting influx, it has the efflux, it has a replenishing reaction. So this is all there here. And I just want to quickly show the video where you see multiple droplets. They tend to have the same size. Obviously, there's much more going on. In particular, in Job's system, there's gravity, which I can simply disable in my simulations. But of course, in the lab, that's much harder. And that's why these droplets fall down. But you can read this publication and convince yourself that this falls exactly in this class of systems that I was describing. So let's basically briefly stop here and take note of what we already discussed and what we achieved. And I'm still maintaining that one can think of droplets as a simple form of life. I showed you that one can view chemical reactions as some simple form of metabolism. there's some form of homeostasis, in particular with regard to the droplet size. Just showed you that. And then there is some growth and change. You can easily imagine that if I change the amount of metabolites in the surrounding, then droplet size also adapts to this. I gave you an equation which says that the droplet size depends on the divisivity and reaction rates and so on. And this is one very simple form of growth and change and therefore development. So there are three other points that I still want to go through. And I want to do this in this order. So in order for this idea of droplets being a simple form of life to have any merit, we also need reproduction. And miraculously, the same equation that I showed before, which has a maintained droplet size, also has an interesting shape instability, where when you start in a box, you're in a 3D box with the small droplets, which is slightly deformed, you can actually go through cycles of growth and division, like so. This is really just the same numerical simulation now in 3D, because it turns out that in 2D the division wouldn't happen. And this clearly looks a lot like cells dividing already, mainly because I colored this green, but that's a detail. But you still see the division. And I can also tell you where this division comes from. There are basically two terms that compete for one another. On the one hand, we have the surface tension driven effect, which basically wants droplets to stay around. That's something I introduced already. But on the other hand, we now, with this chemical reaction, also have continuous influx of material from the surrounding. And then if you have a deformed droplet like so, you can imagine that the influx at the pointed ends is a bit higher than at the blunt ends. I tried to illustrate this with these contour lines. And for those of you who have taken electrostatics, they might remember that the electrostatic field actually also has field lines more closely to each other at pointy ends, like antenna also. And that's exactly the same physics here, because in the end, it's also just a Laplace equation, which governs here diffusion, and electrostatics, the electrostatic potential. So what this means is that there's a higher influx at the pointed ends, so droplets grow there a bit more as compared to the blunt ends, and this, because material is also taken away, in the end leads to spontaneous division. So we understand this. For the experts in the group, this is a little bit related to the Malin-Zakerke instability, albeit we really need this to have this pinch off. We also need this metabolism inside the droplet, which makes droplets shrink. Now, in this particular case, you can also draw another interesting analogy with life, which becomes apparent when you look at the state diagram of the system. So here I have three different phases as a function of the parameters, where it shows the reaction inside and outside as axes. And then I have regions where there are no droplets, just stable droplets or dividing droplets. And if I just relabel these physical parameters of the reaction strength with something more biological, for instance, how much I replenish has to do with how many nutrients are available, whereas how strong the reaction runs inside the droplet is what I called metabolism earlier. And if I play this goal, I can basically go from a droplet, and then if the nutrient availability increases in the environment, I go to the right, things divide spontaneously, whereas if nutrients go away, droplet dies. So I think that these kind of games already sound a lot like simple games of life. So I think we have shown reproduction, and I'd like to move on and rush through this really and talk about response to environmental stimuli. And here I'd like to bring one interesting example of such chemically active droplets that have been discovered in cells. That was in the time when I was interested in Tony Hyman's lab, and Cliff Brangwyn, who's now a professor at Princeton, was a postdoc at the time. And he looked at these little droplets called P granules that you see in yellow here, which are very dynamic. And in this particular example of a developing C. elegans embryo, these P granules all end up on one side. And the reason this is exactly because these droplets respond to environmental cues, which in this example come in the form of a concentration gradient of another protein known as MAX5. So what I'm showing in this example from biology is that droplets really react to gradients, really gradients of all kinds of form. And something we are looking into in a bit more detail now is how droplets respond to diffusive gradients, meaning that material enters the droplet a bit more strongly on one side than on the other. You can then imagine that the droplet responds by moving like so. Or even more relevant, I think, you can also think of hydrodynamic flows, or real flows in the surrounding. And these can be generated by gradients in surface tension, known as Marangoni flows in the end. And then in a simulation, which is carried out by a PhD student in the group, you can see how this droplet can then swim along gradients. So these are just examples of how droplets react to gradients. And I want to bring one more experimental example to show that this is not just a theoretical idea. This comes from our collaborator, Eric Dufresne at Cornell, who reconstitutes droplets in vitro, running a chemical reaction inside, much like I was showing here. And this reaction actually generates gradient around the droplets. These gradients then are felt by the droplets, so there's a whole feedback going on, and you can see the violent flows inside these droplets from these tracer particles. So this is just, we don't fully understand the system. There's a lot going on in the background. This is also from last year, but I think it's a really interesting, 2 years ago, this is really interesting playground to explore more physics. Now, the final example from our group that I want to mention goes back to this idea that we have diffusion and conversion. And here I have a situation where I have two species, which I call solvent 1 and solvent 2, which are converted into each other in much the same manner than we discussed before. And now we have a third species, which actually phase separates from these two. So you have the concentration profile as a function of space. We have phase separation between the two solvents and this solid, and the solvents convert into each other. Let me skip over some of the math, which is not so important, just show you some videos. In particular, if I have such a situation at another species, now I have 3 species coexisting, conversion between the solvents, then I can get interesting dynamics where even without hydrodynamics, I have a well-defined droplet size, much like we showed before, and it can move in particular directions. So here it's actually invaginated here into this large solid phase A. And I can also do the opposite. I can have this droplet basically pinch off from a microscopic phase. So this is just an example, but it shows that with this chemical drive, this metabolism, you can actually also basically make swimming droplets and not just control the size. So it's clear that the droplets would generically respond to environmental cues. And so what this leaves us with is evolution as a last part. And here I must say I haven't done any work myself, but I'd like to highlight one work by my friend Christoph Weber. He's a professor at Augsburg University, and he works in collaboration with Max Planck fellow Dieter Braun in Munich. And they looked at actually in this case, theoretical work where the molecules that phase separate are basically polymers, heteropolymers, in this case, made out of 2 different kinds of monomers. So you can think of these as 2 bases if you want to think about the information encoding here. And then by combining standard physical processes like oligomerization, partitioning, and fragmentation, he could show that particular sequences are very much enriched in these droplets as compared to others, which already gives you an idea that these droplets can carry identity by the sequence of the polymers, and also there's a feedback on the of the polymers on which droplets survive. And so this gives us the first idea of how evolution could be realized in these very simple systems. But I should stress that we are just at the beginning of understanding this. So in the end, I would put a question mark at this. So there's still a lot of research to be done in this direction. Now, just to wrap up, I think I really ran through some ideas that we are toying with in the group, all under the umbrella theme of thinking of droplets as a simple form of life and thinking what they can do, how they can respond to the environment, and what's really possible in these driven systems where we have chemical reactions that break the normal tendency of passive systems. Now, I should stress that this is just one way of seeing all this. We use the very same physical techniques to also understand how droplets actually play a role in organizing the intracellular environment of cells. And I'd like to leave you with this idea that droplets may not only play a role in the primordial life, but also we know for sure that they play a big role in organizing nowadays cells. And I'd like to highlight this review that we published last year, where many of the ideas I was rushing through and some more are explained with, well, a lot more detail and then many other references, in particular from experimental situations. So with that, I'd like to thank you for your attention, and I'm, of course, very happy to take questions.
So, Thank you so much for this illuminating talk that showed that droplets can actually be equated to life. So for those attending, please enter your question in the Q&A and we will read it out and we would address it.
I should say that the whole theme is a bit provocative on purpose. I'm not claiming that this is how life emerged, because obviously I have no idea. But I think provocative ideas are helpful for stimulating and starting a discussion. So,
I have two questions, if I may. The first one is, would you then classify viruses as also life?
Here's my way out. I don't care whether you call them life or not. What I would be interested in is a quantification, right? I mean, along these 6 axes or dimensions that I introduced, or maybe others that you favor, you can quantify how much viruses, resemble life. And then I would turn it into a quantitative question. Maybe you would then come to the conclusion that viruses are 60% alive according to your quantification.
Well, answered like a true theoretical physician.
No, it's maybe my pet peeve, but I'm a big fan of turning qualitative questions, which are hard to answer, into quantitative questions, which you can measure. And I think that's not just for theorists, but also for experimentalists. Otherwise, it becomes semantic discussions.
Okay. My other question was when you discussed about, you try to recapitulate life in these droplets. Do they still behave the same way when you add multiple influxes and multiple components into the same droplet? Do they still, for example, do division? Do they have Oswald ripening? Do they follow the same rules, or does it become more complicated?
Well, you're absolutely right. I mean, it certainly becomes more complicated, and we are only starting to begin understanding what the rules are and what we would expect. It certainly also depends on the details, like how you run the reactions. There are a lot of details that are skipped over for the lack of time. Even in the simple case of just a binary droplet, there's lots more to say. We are now, like one thread in the group is actually pushing toward multi-component systems much more. And well, what can I say? What I learned in the last year is that these are much more complex, much more complicated. And we don't have conclusive answers right now. But I think there's a large room for interesting dynamics. Ours and others group, for instance, have also shown oscillations and more complex dynamics that can emerge once you expand the space and the complexity. But who knows what else will be there? And that's really an active thread of research. Well,
hopefully the next lecture series.
Maybe.
So we have questions from the audience too. From Jed, is the tendency of droplets to reach equilibrium a kind of homeostasis?
It's a very interesting question. I haven't thought about this in this way. I mean, in the end, it depends on what you call homeostasis. The way I understand it, and people might disagree, is that it's actually a property of a system, be it a cell or in my case a droplet, to maintain a certain state independent or like in phase of fluctuations of the environment. And in that sense, I would argue that the normal tendency of a passive system, an equilibrium phase separation, would not be homeostasis, because it's basically completely at mercy of changes in the environment. If I add more material, the thing will grow. I can't really do anything. I can't buffer any of these fluctuations. And so in that sense, I would say homeostasis is something different. It's rather a property that requires non-equilibrium processes. But that's just my take.
Okay, the next question is along the same lines from Davey, who thanks you for the talk. And is the dividing droplets transition to homeostasis, is it because of volume becoming saturated, or does it occur due to reduction of nutrient concentration?
Very good question. Very good observation. Let me show the movie again. So we started here with a situation where we only have a single droplet in this simulation box, which is outlined by these lines. And then what happens is that the droplet basically grows beyond a certain ratio of a certain size where it becomes unstable. But you have very well aptly remarked that if this division would go on forever, then we would fill the box, right? And eventually what will happen actually is that these droplets compete for the material in the surrounding, meaning that they can't grow as big, and it would never cross this instability threshold. So what happens in the long term in the system is that it divides until there are so many droplets that each individual droplet is small enough to be stable. And the system would then basically not show any dynamics anymore. However, there would still be this continuous turnover of material that actually leads to the stable droplet size in the first place, which I mentioned earlier.
Okay, the next question is from Anirudh. Do you think phase-separated droplets could have provided a primitive organizational framework during eukaryogenesis that reduced the selective pressure for immediate membrane enclosure, thereby influencing how and when membrane systems evolved?
It's a super interesting question, of course.
Yes.
Unfortunately, I mean, I can't make any definite statements about what happened at the origin of life, as I said before. But I think it's intellectually interesting to think about alternatives, because I think the prevalent picture is that we had something like a membrane vesicle with a well-defined lipid membrane, and then all the additional machinery of life was developed afterwards. Now, what I'm putting forward here is this picture, which, as you observed, doesn't need any membrane, and can achieve, as I would claim, many of the relevant aspects of life. Now, I think both these views are extremes and therefore probably both wrong. And what I would rather expect is that life started somewhere in between with a much more messy situation, where maybe some of the organization was due to phase separation, as I was saying here, but surely there would be molecules like dirt molecules sitting on the surface, which could have very well been lipid molecules. But then it would be a messy membrane with lots of holes and so on. But it might have fulfilled some of the roles that membranes are certainly necessary for nowadays, be it to restrict what goes over the interface, or also provide mechanical stability and so on. So I think that, well, life used all these physical possibilities, and I wouldn't be surprised if some intermediate picture was actually the true answer, although it's hard to tell, obviously. That's my subjective view on this.
The next question is from Mauro, who would like you to clarify how the efflux of chemicals outside of a chemically active droplet scales with the volume of the droplet. Once the waste is produced, shouldn't its flux from the inside to the outside of the droplet also depend on the surface of the droplet, as well as the diffusion of waste within the droplet?
Yeah, I mean, I went over this very, very quickly, and there are lots of details. In particular, these scalings that you presumably refer to, show here on the slide, are scalings that only occur in the limiting case of some simplifying assumptions I used. However, the observation, namely that the growth reaches a stable size, that holds for more general assumptions, because all you need is that the influx scales weaker with the radius than the efflux. Now, let me try to elucidate a little bit more clearly where these scalings come from. And they really rely on what I would call a length scale separation, where the droplet is very small to the environment. And diffusion is rather fast, reactions are rather slow. And in this situation, I have a well-defined droplet. And because the reactions are slow, the material from the outside comes from a large environment surrounding the droplet. And how much arrives at the droplet is really limited by diffusion. Reactions don't play such a big role besides setting up a supersaturation. And then to get this R scaling here, you basically just solve the diffusion problem onto a sphere. You will find, if you do this, it's basically the Laplace equation, you find that locally the diffusion is proportional to the inverse of the radius. You integrate this over the surface area, which gives you a radius squared, which together implies that the integrated flux over the surface scales like the radius. Now, for the F flux, we're in a very different situation because we have a droplet, which in this case, where reactions are slow, is rather homogeneous. And then this, what I call metabolic reaction, this conversion of droplet material to base, runs everywhere in the volume at roughly equal rate. And once I created a waste molecule inside the droplet, it actually wants to leave because it doesn't like the droplet phase. That's the underlying assumption here. And so the flux with which this leaves is in this case actually not limited by diffusion because the droplet is so small. It's limited by the small reaction going on inside the droplet. And that's why I can estimate this flux by the reactive flux, which is simply the reaction rate times the droplet concentration times its volume. Now, these are limiting cases which make this argument a bit simpler. Realistically, scalings will be here probably between r and r squared, and here between r squared and r cubed, but you would qualitatively get still this behavior. That's the key.
Okay. The next question is from Krisha. In the videos depicting the yellow droplets interacting, the primary central droplet both increases and decreases in size. What would determine the direction of influx or efflux, which occurs between the two droplets?
Are you talking about this video?
It says yellow droplets interact. Yes.
Yeah, yes, good. Okay, so the majority of my talk was about theory and nice, clean schematics where I know exactly what's going on. The difference to this talk, this slide, is that this is messy experiments where things are much more complicated. But in brief, and I've not worked on this. I mean, this is again Christoph Weber from Augsburg and Jörg Bölkhofen from Munich. But what I learned from the system is that they indeed have such a reaction cycle as depicted in the schematic taken from their publication. And they basically have a system which is indicated by this white circle here that you should think of as a closed system where phase separation happens, and phase separation gives rise to these yellow droplets. And however, because there's gravity and these droplets are denser than the surrounding medium, they fall down. And in principle, if there were no reactions, they would form a large droplet at the bottom of the system. However, because there is this deactivation reaction, which is the simple form of metabolism in my language, this droplet would actually shrink a little bit. And you can see that it doesn't grow, it actually shrinks continuously, giving rise to, in my language, waste products, which diffuse in the background in the entire system and actually give rise to nucleation of droplets up here because there's this reactivation reaction happening. And then new droplets form, they grow, and then they emerge here. Now, again, because this is an experiment, there's much more going on. And if you watch closely, you can even see that this falling droplets are not falling continuously, but they're like waves of droplets with a well-defined oscillatory period. And well, if you want to know more about this, you really have to read this very nice paper by my colleagues. But the main idea is that this is really a system where droplet size is controlled by such a activation-deactivation cycle. But on top of this, there's additional things going on, which the experimentalists simply couldn't disable to have a cleaner system. And so we have to deal with that. But yeah, that's the best we have so far.
Well, it's neat theory meets messy practicals.
Yeah, that happens. I'm fine with that.
Yeah, I think we have answered all the questions. There are no more. So with that, I would thank you once again, David, for taking the time to explain your research, to explain these really nice concepts that all of us can understand, and also for answering all our questions.
It was a pleasure. Thank you very much.
So thank you to all the participants, all the attendees for joining us. And we will, if you're interested, the next lecture series will take place in February, and it would be in the next MtL Life Fellow, Serafina Wegner. And you'll find information about it on our social media and on our website. Thank you so much. Have a nice evening, morning, wherever you are, whatever time it is.
Thank you for showing up.